Welcome to this subunit on regeneration of samples of plant genetic resources. This subunit is part of Unit 1.1 Plant Genetic Resources within Module M1 Biodiversity and Agro-Resources. This is a session regeneration of samples of plant genetic resources. In addition to this video, this subunit includes a PDF file on the Biodiversity International Regeneration Guidelines for Plant Germplasm. By the end of this course, you will be able to determine when an accession has to be regenerated, to design a germplasm regeneration program, and to select between different strategies for the regeneration of allogamous species. The subunit is divided into sections. A first one stressing that conservation of germplasm is linked to regeneration, and the second one emphasizing that regeneration is linked to the reproductive system. After germplasm is collected, accessions are normally regenerated. Also, regeneration takes place after a certain time when viability has decreased or when the seed stock is reduced. Some important issues to take into consideration in germplasm regeneration are the following. When should an accession be regenerated? What seed should be used, the original or regenerated one? How is the regenerated seed identified in the collection? What is the structure of variation within and among accessions in the collection of a crop or wild relative? And this largely depends on the reproductive system, and particularly influences the number of plants of accession to be used, the number of individuals or seeds to ensure the genetic integrity in each cycle. The pollination methods depend on the reproductive system. The use of pollinators can also be used in certain cases in allogamous crops, and spatial isolation may also be needed to avoid genetic contamination in allogamous crops. In order to perform the regeneration of germplasm, certain guidelines should be followed. The basic guidelines are attached as a file of this subunit. The genetic structure of crops is linked to the reproductive system, and therefore regeneration has to take this into account. In autogamous species, such as tomato, the plants self-themselves and facilitate the regeneration of germplasm. In partially allogamous species or partially autogamous species, a special caution has to be taken to avoid cross-contamination with pollen from other accessions. In allogamous crops, during regeneration, we normally will stimulate cross-pollination among individuals of the same accession to avoid inbreeding depression, but also special measures like mesh cages need to be taken to avoid cross-pollination with other accessions of the same crop. So, for allogamous species, manual cross-pollination is an alternative to ensure that there are cross-pollinations. Here we have an example of artificial cross-pollination from melon, which is an andromonoecious species. This means that the plant has hermaphrodite and male flowers on the same plant. Alternative to ensure cross-pollination in allogamous crops is the so-called chain pollination, in which one plant serves as male for the next plant in the row. In some cases, there are species which are self-incompatible and therefore are necessarily allogamous, like some wild relatives of tomato. The alternative in this case is to make a mixture of pollen of several plants and make manual pollinations using this mixture. A common problem also in allogamous crops is that sometimes there are off-types within one accession. In this case, tagging the crosses may help in selecting only the crosses among plants of the same type, ensuring the maintenance of the integrity of the accession. The most important messages of this subunit are: first, regeneration of germplasm is needed when an accession is collected or when seed stocks are low. Second, it is important to follow established regeneration guidelines to ensure that the accession conserves its integrity and does not suffer genetic contamination. Third, the methods for regenerating germplasm depend on the reproductive system. Fourth, in autogamous species, the plants can be left to self-pollinate, ensuring that the offspring is genetically identical to the mother plant. Fifth, in allogamous crops, cross-pollination has to be stimulated, and this can be done using different systems. And sixth, tagging of crosses in allogamous crops can allow detecting crosses within accession with off types that should be removed.